Good day, everyone. Once again, we are the Group Orange of BSCHEM 31, and we are here to discuss to you the general biochemical cycles that occurs in photosynthetic eukaryotes, specifically the plants. So to give you a brief introduction to photosynthesis, as well as the structure chloroplast, where the photosynthesis process occurs, we have Miss Albornoz. The light and darker actions of photosynthesis will be discussed by Ms. Arce and Mr. Billonid. And finally, the plant metabolism, Ms. Reyes will tackle the photorespiration, which commonly occurs in C3 plants. Ms. Martinez will tackle the general photosynthesis process in C4 and CAM plants. And lastly, Mr. Terrado will discuss the integration of general metabolic processes in plants, the photosynthesis, and the cellular respiration.
I am Gabrieli Monique Albornoz, and I will be discussing about a brief introduction to photosynthesis and why is it important, as well as discuss the chloroplast structure of plants. To begin, photosynthesis in layman's term is a natural process of food making in plants. It is a biological process that involves the use of sunlight, water, and carbon dioxide to produce oxygen and energy in the form of sugar. As we all know, plants are one of the most important living things in the planet. We depend on them for our daily survival, including food, water, and habitat. The process of photosynthesis makes those things happen. It helps balance our ecosystem by using solar energy and transforming it into something useful and beneficial, not just for plants, but to other organisms as well. Chloroplast is a key factor in conducting photosynthesis. It contains chlorophyll, which is responsible for absorbing sunlight and turning it into useful energy. The chloroplasts are cell organelles that are found only in plant cells. It is composed of two membranes identified as the outer and inner membranes. Inside that membrane is the stroma. This is where the Calvin cycle takes place. It is a protein-rich fluid surrounding the stalks of thylakoid called the grana. Chlorophyll are found inside the thylakoid membranes, and this is where the light-dependent reactions takes place. During this process, two molecules, namely carbon dioxide and water, act as the reactants. Electrons are transferred from water to carbon dioxide, and the chlorophyll absorbs the light energy and electrons become excited from lower energy state to higher energy state. This will be further discussed throughout the video.
As said a while ago, the photosynthesis undergoes a complex series of enzymatic reactions to synthesize sugar and oxygen gas. This will be done from the basic compounds like water, carbon dioxide, and using the light energy source from the sun. Actually, the photosynthesis of the plants can be divided into two stages of reaction cycles, which namely are the light cycle and the dark cycle, which is also known as the Calvin-Benson cycle. So I am Meros Arce, and I will discuss to you the part 1 of photosynthesis, which is the light cycle. Obviously, in its name, the light cycles is light dependent and its light source is the sunlight. The light cycle also uses water, and through the oxidation of this water, it produces oxygen gas molecule. The light cycle also produces NADPH and ATP. This NADPH and ATP are important since this will be used later for the dark cycle or the Calvin cycle. And the light cycle takes place in the granum within the chloroplast, so here, in the thylakoid membranes. So we have two types of photophosphorylation in the light cycle. First one is through the non-cyclic electron transport, which can we call the non-cyclic photophosphorylation. And the second one is through the cyclic electron transport, which can we call cyclic photophosphorylation. But before we go to the main discussion, let me introduce to you the photosystem 2 and photosystem 1. These two are multi-protein complexes that contain pigments that are necessary to harvest photons. And those photons will be used to catalyze the primary photosynthetic and organic compounds, which leads to the production of high-energy compounds. Also, this photosystem 2 is called P680 since it absorbs light at maximum absorption wavelength of 680 nanometers, while the photosystem 1 is also called P700 since its maximum absorption wavelength of light is at 700 nanometers. So going here to the non-cyclic photophosphorylation, this process is mainly divided into two parts, which is the non-cyclic electron transport and the chemiosmosis, which causes the photophosphorylation. Let us focus first the non-cyclic electron transport. The photosystem 2 contains chlorophyll, which has immobilized electrons. And when these immobilized electrons are struck by a light or photon, this becomes excited and flows out through the photosystem and goes to the plastophenone, which is an electron mobile carrier. When the photosystem lost its electrons, this should be replenished by the oxidation of water, which gives out of its electrons to the photosystem to suffice the loss of electrons. And also, the oxidation of water also gives out oxygen gas molecules and two hydrogen ions. So going back here to the electrons flowed in the plastophenone, the plastophenone then delivers the electrons to the complex cytochrome B6F. And as the electrons passes through the cytochrome B6F, the cytochrome B6F pumps in the protons from the stroma to the thylakoid lumen or interior, which causes a proton gradient. And after the electrons have passed through the cytochrome B6F complex, this goes to the plastocyanin, which is a copper-containing protein. And from plastocyanin, it is then transferred to the photosystem 1. And at this point, the electrons have already lost some of its energy. And by striking another photon, the electron becomes more excited and it will flow to the pyridoxine, which is an iron sulfur protein. From pyridoxine, this will be transferred to the pyridoxine NAD pyridoxase, and the electron transferred to this enzyme aids to reduce the NADP to become NADPH. As you can see here, it uses a proton, which again lowers the concentration of protons in the stroma, and thus the gap of the gradient of the thylakoid interior and the stroma increases. And due to gradient, the production of ATP will become favorable. And as the thylakoid interior has excessive protons, the process of chemiosmosis will now occur through ATP synthase. The protons will be pumped out from the thylakoid interior to the stroma, and as the ATP synthase pumps out protons, the ATP is phosphorylized to ATP. So overall, the products for the non-cyclic photophosphorylation are the oxygen coming from the oxidation of water. Next is the ATP, which is phosphorylated from ADP through ATP synthase. And the third product is the NADPH, which is reduced from the pyridoxine NADP reductase. In anabolic processes like the Calvin cycle, when we compare the usage of ATP and NADPH, the ATP is more commonly used than the NADPH. Thus, there are tendencies that the production of NADPH becomes excessive. So, how do we stop the production of NADPH? This can be done through the cyclic photophosphorylation. where the electrons from the photosystem 1 does not proceed in the pyridoxine NADP reductase and just reverts back to the photosystem 1 cyclically, and it repeats over and over. And same scenario with the non-cyclic photophosphorylation when the protons inside the thylakoid interior becomes excessive due to the pumping in of protons, the chemical osmosis occurs, which causes to photophosphorylate the ATP to ATP through the ATP synthase. So in cyclic photophosphorylation, only the ATP is the product, and only the photosystem 1 is involved in this process. So in summary, if we will compare the non-cyclic and cyclic photophosphorylation, both the photosystem 1 and photosystem 2 are involved in the non-cyclic photophosphorylation, while for the cyclic photophosphorylation, only photosystem 1 is involved. In non-cyclic, P680 is the active reaction center, while for the cyclic photophosphorylation, P700 is the reaction center. So for the non-cyclic photophosphorylation, obviously it travels in a non-cyclic manner, while for the cyclic photophosphorylation, the process just repeats itself, so it behaves in a cyclic manner. So for the non-cyclic photophosphorylation, the electrons from photosystem 1 are accepted by NADP plus reductase. While for the cyclic photophosphorylation, electrons just reverts back to the photosystem 1, and the electrons is not accepted, or it does not proceed to the NADP reductase. For the non-cyclic photophosphorylation, both NADPH and ATP molecules are produced. And for the cyclic photophosphorylation, only ATP molecules are produced. For the non-cyclic photophosphorylation, since the photosystem 2 is involved, the photolysis of water is present, and oxygen is one of the byproduct. While for the cyclic photophosphorylation, since the photosystem 2 is not involved, water is not required in the process. And lastly, for the non-cyclic photophosphorylation, this process is predominant in all green plants, while the cyclic photophosphorylation is predominant only in bacteria. If we will summarize the whole light-dependent reaction or the light cycle, its reactants will be the water, the light, the NADP plus, the ADP, the phosphate, which yields the products oxygen gas molecule, NADPH, and ATP that will be used for Calvin cycle that will be reported by John Mark Billionid.
Good day, everyone. I am John Mark Ibilionid, and I will be the one to discuss the Calvin-Benson cycle. So first, what is Calvin-Benson cycle? So it is the stage 2 of photosynthesis pertaining to the light-independent or dark reaction. Here in Calvin-Benson cycle, the energy equivalents ATP and NADPH, which are derived from light reactions, together with carbon dioxide, will be used to produce sugars such as glucose, which is the main energy source in many organisms. Calvin-Benson cycle has 3 steps. First one is the carbon fixation. The second one is the reduction and production of sugars. And the third one is the regeneration of RUBP. Calvin-Benson cycle occurs in the stroma of chloroplast organelle inside the plant cell. So without further ado, let's start. So this is what I'm referring to. So just a quick recap, the light reaction occurs in the thylakoid membrane inside the chloroplast, while the Calvin cycle occurs in the fluidic space, which is the stroma, also inside the chloroplast. So to begin with, we have 6 moles of ribulose 1,5-bisphosphate, and together with 6 moles of carbon dioxide obtained from the environment, it will form 6 moles of short-living intermediate, which consists of 6 carbon atoms. Now, the enzyme that catalyzes carbon fixation is no other than Rubisco. And Rubisco stands for RUBP carboxylase oxygenase. Here in Calvin cycle, Rubisco specifically acts as carboxylase. You will see in the later discussions that Rubisco can also act as oxygenase depending on the situation. Now, Rubisco is the rate-limiting enzyme in photosynthetic carbon fixation. It is one of the most important enzymes in a biosphere. In fact, it is one of the most abundant enzyme in our planet. Without Rubisco, photosynthetic organisms such as plant won't be able to catalyze carbon dioxide and consume energy equivalents to produce glucose and other sugars. Now, going back to our discussion of Calvin cycle, we have 6 moles of short-living intermediate which rapidly degrades into 12 moles of 3-phosphoglycerate. Now, the phosphorylation of this intermediate to 1,3-bis-phosphoglycerate needs consumption of 12 ATP. Now, 12 moles of 1,3-bis-phosphoglycerate will be reduced to 12 moles of glyceraldehyde 3-phosphate. Since it's a reduction, it needs 12 NADPH, which is a reducing power. Now, the glyceraldehyde 3-phosphate has two fates. 2 moles of glyceraldehyde 3-phosphate will be used to synthesize carbohydrates or sugars, specifically in our example, the glucose, which contains 6 carbon atoms. And the remaining 10 moles of glyceraldehyde 3-phosphate will be used to regenerate 6 moles of ribulose 1,5-bisphosphate. And in this process, it has to consume 6 ATP. So the Calvin cycle will perpetuate again and again. So here's the overall reaction of the Calvin cycle. So we have 6 carbon dioxide from the environment together with 6 RUBP and the consumption of 18 ATP, 12 NADPH, and 12 H plus will yield 2 moles of glyceraldehyde 3-phosphate, 6 RUBP will be regenerated, 18 ADP, 18 orthophosphate, 12 NADP plus, and another 12 orthophosphate coming from the dephosphorylation of 1,3-BPG to G3P. And these 2 moles of glyceraldehyde 3-phosphate will be used to synthesize 1 mole of glucose. And glucose, as we all know, is the main energy source in many organisms, which will be further oxidized to yield ATP, the main energy currency inside the biochemical system. Now, this is what I want you to notice in the Calvin cycle. So first thing is that it is analogous to citric acid cycle. So upon the addition of carbon dioxide, there is no net addition of carbon atoms. So what happens? What happens is that the carbon atoms contributed by carbon dioxide will just be used to synthesize carbohydrates, in this case, the glucose. And the remaining carbon-containing intermediates will just be regenerated for the Calvin cycle to perpetuate. Now, second thing, can you notice these 3 intermediates? So let me encircle this for you. Yeah, this is this one. So I'm referring to these 3 intermediate. We can also find this in glycolysis. In Calvin cycle, the direction of reaction is clockwise. That is why it consumes energy equivalents. On the other hand, in glycolysis, the direction of reaction is just the reverse, which is counterclockwise. That is why in glycolysis, we have yielding of energy. And last thing that I want you to notice is that the first product of carbon fixation in Calvin cycle is a 3-carbon intermediate, which is 3-phosphoglycerate. That is why Calvin cycle is also called as C3 cycle. And Calvin cycle, take note, it occurs in all plants. It occurs in all plants. The only difference of C3 plants in C4 and CAM plants is that the C4 and CAM plants has additional mechanisms which makes them more efficient in terms of carbon fixation. So before I end my discussion in Calvin cycle, let me introduce to you briefly the concept of photorespiration. In C3 plants, photorespiration commonly occurs. So this is what I'm telling a while ago that rubisco can also act as oxygenase. And in photorespiration, rubisco is the enzyme that is also used, and for that purpose, rubisco acts as oxygenate. So I will not be discussing it in detail since it will be further tackled by Miss Reyes. So that's all for my discussion in Calvin cycle, and thank you for listening.
Hello, my name is Grace Reyes, and I'm going to report about plant metabolism photorespiration. So first, what is photorespiration? According to Merriam-Webster, photorespiration is defined as a light-dependent process in some plants resulting in the oxidation of glycolic acid and release of carbon dioxide that under some environmental conditions such as high temperature tends to inhibit photosynthesis. In other words, it is a process found in normal C3 plant respiration. Here, the enzyme rubisco uses O2 as a substrate instead of carbon dioxide. This alternative pathway leads to the loss of fixed carbon, wastes energy, and decreases sugar synthesis. By the way, they are called C3 plants because of the 3-carbon molecule 3-PG. So there are 3 factors that affect photorespiration. First, we have temperature. Increasing temperature also increases our oxygenation in 2 ways. First, rubisco loses affinity for CO2 as temperature rises such that it performs oxygenation. The second possible way temperature affects oxygenation is by its effect on the relative solubilities of O2 and CO2. Both the gases must dissolve in the water of the leaf before they can react with the rubisco. With the increase in temperature, the solubility of any gas in water decreases. There is comparably less O2 and CO2 dissolved in cells at higher temperature. However, the solubility of O2 decreases less with temperature than CO2. Thus, as the temperature rises, the CO2 to O2 ratio in the cell decreases, which starts favoring the oxygenation of rubisco. Next is moisture content. As the moisture content in the surroundings decreases, plants close their stomata. Inside the leaf, O2 is produced and starts to increase its concentration. At the same time, CO2 is consumed by carboxylation and thus decreases in concentration. Consequently, CO2 to O2 ratio inside the leaf decreases when stomata are closed, which promotes oxygenation of rubisco. Thus, hot and dry conditions stimulate rubisco to perform photorespiration in plants by increasing its affinity for O2. So again, both these factors result in the relative concentrations of O2 and CO2, which gives us the third factor. So what exactly happens during photorespiration? Photorespiration begins in the chloroplast of a C3 plant when rubisco uses O2 and RUBP as substrates instead of CO2, producing one molecule of 3-PG or 2-phosphoglycerate and one molecule of 2-PG or 2-phosphoglycolate. The equation is shown in the screen. So the 2-PG is inhibitory to the Calvin cycle enzymes and thus needs to be removed and recycled back to 4-PG and to prevent depletion of Calvin cycle intermediates. This recycling is complex and involves 3 different organelles, which are chloroplast, peroxisome, and mitochondria. The 2-PG formed in a chloroplast by the oxygenase activity of rubisco here is first dephosphorylated to glycolate. Glycolate is then transported from the chloroplast to the peroxisome. Here, the peroxisome. It is then metabolized to glyoxylate.
by the enzyme glycolate oxidase. This reaction consumes O2 and generates toxic molecule H2O2 and later detoxified by the enzyme catalase, regenerating O2. Then the two glyoxylate molecules are converted to glycine in a transamination reaction with glutamate acting as amino donor for one glycine and serine as an amino donor for the second glycine. Glycine formed is transported to the mitochondrion here, where one glycine molecule is catabolized to ammonia and CO2. This reaction, catabolized by the enzyme glycine decarboxylase complex, adds the remaining glycine to a second glycine molecule to form serine. Serine formed in a mitochondrion is then transported back to the peroxisome. It then undergoes transamination with the amino portion serving as an amino donor for the second glycine and the remaining carbon forming hydroxypyruvate. Pyruvate is then reduced to glycerate, exported to the peroxisome, the chloroplast. They are then phosphorylated to form 3-phosphoglycerate or 3PG here, which can re-enter the Calvin cycle. This pathway is called photorespiration because it consumes O2 here and releases CO2 here. It uses ATP and NADPH produced in the light reactions, just like the Calvin-Benson cycle or the Calvin cycle for short. And so going back to the organelles responsible for photorespiration, we have chloroplasts, peroxisomes, and mitochondria, which were already discussed in the previous slide. This one is just the summary. So next we have the consequences of photorespiration. A disadvantage of photorespiration is that one, it is bad for plants. Photorespiration is thus two, a wasteful pathway because it significantly lowers the efficiency of photosynthesis and thus, number three, decreases the productivity of plants like wheat and soybean. It does so by decreasing the efficiency of photosynthesis and by preventing them from using ATP and NADPH during the carbohydrate synthesis. It also results in number four, a loss of 3 fixed carbon atoms. So how does Rubisco decide whether to act as an oxygenase or as carboxylase? This one is related to the earlier discussion, the factors affecting photorespiration. First, Rubisco has 10 times more affinity for carbon dioxide and O2 and so favors CO2 fixation. Another consideration is the relative concentrations of CO2 and O2 in the leaf. If O2 is relatively abundant, Rubisco acts as an oxygenase. And photorespiration ensues. If CO2 predominates, Rubisco fixes it, and the Calvin-Benson cycle occurs. Temperature is also a factor. As mentioned before, photorespiration is more likely to occur at high temperatures. On a hot, dry day, the stomata that allows water to evaporate from the leaf closes to prevent water loss, but this also prevents gases from entering and leaving the leaf. The carbon dioxide concentration in the leaf falls because CO2 is being used up by the light-driven photosynthetic reactions, and the O2 concentration rises because of these same reactions. As the ratio of CO2 to O2 in the leaf falls, the reaction of Rubisco with O2 is favored and photorespiration again proceeds. That is all from my report and thank you for listening.
Previously, the photorespiration of C3 plants was discussed. In this part of the presentation, we will talk about the photorespiration of the C4 and CAM plants, as well as its general pathway, examples, their differences, and efficiency. First, let us discuss what is C4 cycle. C4 cycle is also known as the Hatch and Slack cycle. The plants that uses this pathway is called C4 plants. C4 plants avoids or minimize photorespiration and uses photosynthesis to continue making sugars. The C4 cycle occurs in mesophyll cells. The pathway works by initially fixing carbon dioxide in the mesophyll cells via the enzyme PEP carboxylase. This reaction reacts the 3-carbon phosphoenol pyruvate with carbon dioxide, resulting to 4 carbon oxaloacetic acid, or OAA. The 4-carbon compound that were in the mesophyll cells then cycles into the bundle sheath cells and releases carbon dioxide. Moving on, we have the CAM cycle. CAM cycle is also known as the crassulacean acid metabolism. The plants that uses this pathway is called CAM plants. These plants minimize photorespiration and conserve water by only opening their stomata as well as admitting carbon dioxide at night. Similar to C4 plants, CAM plants uses PEP reaction, which proceeds once the carbon dioxide enters the leaves. However, as compared to C4 plants, the resulting organic acid of CAM plants are stored in vacuoles. During the day, since the stomata closes to conserve water, the carbon dioxide storing organic acids are released from the vacuoles of the mesophyll cells into the Calvin cycle. In terms of their pathways, C3 does not have the abundance of PEP carboxylase and bundle sheath cells to avoid photorespiration like C4 and CAM plants. Also, unlike C4 and CAM plants that has 2 steps in their pathways, C3 plants is only a 1-step pathway. The 3 plants also have different leaf anatomy. C3 plants has a typical leaf anatomy, which makes these plants at a disadvantage when in dry and hot environment. C4 requires a Kranz anatomy that is significant to avoid photorespiration, while CAM plants are xeromorphic in order to avoid water loss in very high temperature. Let us now have the examples of C4 and CAM plants. First, we have the examples for C4 plants. We are familiar with these plants such as corn and sugarcane. Other examples including switchgrass, sorghum, and maize are also an example of C4 plants. These plants are more drought resistant. Also, it requires less energy, which make it more efficient compared to C3 plants. Pineapple and cactus, on the other hand, are examples of CAM plants, as well as the snake plant, Apuntia, sedum, and kalanchoe. These plants are extremely drought resistant, and also it is very energy inefficient. When it comes to efficiency, C4 and CAM plants are much efficient than of C3 plants, whose efficiency of the Calvin cycle is reduced due to photorespiration. This is because C4 and CAM plants uses the enzyme PEP carboxylase, which has a higher affinity of carbon dioxide and does not bind to oxygen. Therefore, it prevents the photorespiration from occurring. Thus, C4 and CAM plants are more efficient because of its high rate of photosynthesis and reduced rate of photorespiration. That is all for the photorespiration of C4 and CAM plants.
Good day, everyone. I'm Zenrimi Coterado, and I will be the next discussant. So for this part, I'm going to discuss the relationship of photosynthesis and cellular respiration. Now, we all know that the photosynthesis and cellular respirations are both two biochemical processes, and these two involve in multiple complex steps and share the same molecules such as the oxygen, carbon dioxide, water, glucose, adenosine triphosphate or ATP, and so on and so forth. So in this part, I'm going to discuss the relationship of the two on how they will interact with each other to create an energy cycle in living organisms. So here we have the equation for photosynthesis and cellular respiration. So for the balance equation of cellular respiration, C6H12O6 plus 6O2 yields to 6CO2 plus 6H2O. On the other hand, for the balance equation of the photosynthesis, 6CO2 plus 6H2O yields to C6H12O6 plus 6O2. So here we can notice that the relationship of the two processes, because the product of the one process is the reactant of the other. Notice that the equation for cellular respiration is the direct opposite of the photosynthesis. So in other words, yung product ng photosynthesis are required for cellular respirations, and the product of the cellular respirations can be also used to power photosynthesis, or is also required for photosynthesis. Next is the products of glycolysis and TCA cycle, or the Krebs cycle. The process of glycolysis and TCA cycle, we all know that these two are both involved in cellular respirations. In both of the cellular respiration processes, such as yung aerobic and anaerobic, glycolysis is the first step from which the reaction starts. While the Krebs cycle naman is a metabolic reaction that occurs just after the glycolysis process. So we can say na yung TCA cycle will not happen if the glycolysis will not happen first in the cellular respiration. So for the product of these two, for the glycolysis, we have here the ATP, pyruvate, and NADH, while for the TCA cycle, the product or the output will be the ATP, NADH, and FADH2 as the energy carriers, and CO2 as the waste product. Note that if the Krebs cycle as a part of the cellular respirations makes ATP and NADH, we can associate this in photosynthesis, since na-mention in other reports that the Calvin cycle is part of the light-independent reactions of photosynthesis. So in this case naman sa Calvin cycle, it uses the ATP and NADH. So again, yung Krebs cycle, the output would be the ATP and NADH. While in contrast, sa Calvin cycle, ito naman yung nagiging input. And the same siya na yung input is the ATP and the NADH. Next, lastly, the functions in the organism. So for the photosynthesis, it uses light, water, and carbon dioxide to create food for the organism in the form of sugar, specifically the glucose. While the cellular respiration naman, it uses glucose to make a form of energy that the organism can use in cellular processes, or the adenine triphosphate. So dito, makikita rin natin yung relationship of the two, since the output of the photosynthesis is the sugar or the glucose, while yung input naman is the usage of the glucose for the cellular respiration. So to understand it more further, take a look at the diagram. So here, the photosynthesis makes the glucose that is used in cellular respirations to make adenosine triphosphate. So yung glucose, it turned back into carbon dioxide, which is used to photosynthesis, while water is broken down to form oxygen. During photosynthesis, in cellular respiration, oxygen is combined with hydrogen to form water. It is the released oxygen that is used by us and by the other organism for cellular respiration. So we breathe in the oxygen basically, then which is carried through our blood to all our cells. So in our cell, yung oxygen allows cellular respirations to proceed. So without oxygen, much less ATP will be produced, since yung cellular respirations work best in the presence of oxygen. And additionally, yung cellular respirations and photosynthesis are important parts of the carbon cycle. Yung carbon cycle, it refers to the pathway through which carbon is recycled in the biosphere. So, yung nangyayari dito is yung cellular respiration releases carbon dioxide into the environment. Yung photosynthesis naman pulls carbon dioxide out of the atmosphere. So the exchange of carbon dioxide and oxygen during photosynthesis, as shown in the figure, And the cellular respiration worldwide helps to keep the atmospheric oxygen and the carbon dioxide at stable levels. So in summary, first, the equation for the cellular respiration is the direct opposite of the photosynthesis. Next, the waste product of photosynthesis is used in cellular respiration, and the waste product of cellular respiration is used for fuel and photosynthesis. Lastly, the exchange of carbon dioxide and oxygen through photosynthesis or cellular respiration worldwide helps to keep the atmospheric oxygen and carbon dioxide at stable levels. And that will be all. Thank you.